In this video, I'd like to talk about nucleic acids. Nucleic acids are used to store genetic information. Nucleic acids are important also for the expression of genetic information. There are two types of nucleic acids in your body. One is DNA, or deoxyribonucleic acid. It's used to store genetic information that's used by the cells in your body to control many essential life processes. RNA is ribonucleic acid. It's responsible for carrying out the instructions found in the DNA molecule. And actually, in some viruses, RNA is actually used rather than DNA to store genetic information. You can see here two diagrams. One is of DNA. DNA is actually made of two single strands that are put together. They're complementary strands, which is a term I'll define a little bit later in this video. But they are helical, meaning that they're coiled and they coil around each other. So it's a double helix. And then we have RNA, which is typically found in a single-stranded formation, which you can see here in this diagram. The roles of DNA and RNA can be summarized as follows. DNA contains the instructions for making RNA. RNA contains the instructions for making proteins. And as we've discussed in previous videos, proteins are molecules that carry out important cellular processes. Here in this picture, you can see a DNA molecule. You can see it has this double helical structure, and it's actually been opened up in the middle, and there's an enzyme called RNA polymerase, and it's actually making a complementary strand of RNA. That strand of RNA will actually leave the cell nucleus and will be used to make protein out in the rest of the cell. So DNA and RNA are both polymers. They're composed of monomers called nucleotides. Each nucleotide consists of a 5-carbon sugar. In the case of DNA, the 5-carbon sugar is called deoxyribose. In the case of RNA, the 5-carbon sugar is called ribose. There's also a single or double-stranded structure called a nitrogenous base. It's called a nitrogenous base because it has a lot of nitrogen atoms within it. And it's a base because it is basic. It can accept a hydrogen ion from a solution. And finally, the third part of a nucleotide is the phosphate group, which is a phosphorus atom covalently bonded to 4 oxygen atoms. So it has the chemical formula PO4. There are 4 different types of nucleotides in DNA. Now, note that the sugar and phosphate groups don't vary, but the nitrogenous base does vary. So the 4 nitrogenous bases found in DNA are adenine, thymine, cytosine, and guanine, which you can see in the various diagrams around me. Now let's take a quick look at adenine for a second, just as an example. So we can see how things are bonded together. You have the deoxyribose, which is a 5-carbon sugar. Covalently bonded to that is a phosphate group, PO4. And also covalently bonded to the 5-carbon sugar deoxyribose, you have a nitrogenous base. So again, note the nitrogens within the double ring structure. And it's also basic, as I said. Let's move on. So DNA is normally found in a double helix. Each single strand is formed by covalent bonds between sugar molecules and phosphate groups. So these sugar molecules and phosphate groups essentially make the backbone of each single strand of DNA, and but attached to the backbone, of course, are the nitrogenous bases, and the nitrogenous bases actually face inwards towards the middle of this helical structure. And in the middle of this molecule, the bonds are not covalent bonds; they're hydrogen bonds, which, as we've discussed previously, are weaker bonds than covalent bonds. This means that because these bonds are weak, they're hydrogen bonds, the DNA can actually open up in the middle where the hydrogen bonding occurs. And this opening up of the DNA allows for the genetic code in the DNA to be read and allows for DNA replication to occur. Okay, so nucleic acids store genetic information. The 2 single strands of DNA are said to be complementary. They're complementary because the sequence of one strand perfectly predicts the sequence of the other strand. So let's take an example and look at some of this diagram we have here. So if you see adenine on one single strand of DNA, then the complementary single strand of DNA must have a thymine in the same place, so that the adenine and thymine always hydrogen bond with each other. Similarly, if you have guanine on one single strand of DNA, then you must have cytosine on the other one. And you can see that they also hydrogen bond just with each other. So this turns out that this is very important, this idea of that strands can be complementary. This allows for the DNA, again, to open up, do the hydrogen bonding, and for a complementary strand of RNA to be formed. And also when DNA replication occurs, the hydrogen bonds are weak enough that the DNA can open up, and a complementary strand of DNA can be made, and this allows for the replication of DNA. Okay, that's it for this video. Thanks a lot for watching.